Hello, that's me. How's it going, everybody? I'm going to give you two lectures on immunometabolism. This is a course that Dave Finley put together. I guess this is the third year now, actually, you've done it, and Dave had a great idea. He said, why don't we do a course on immunometabolism for the senior softs? And I said, yeah, fantastic idea. And Dave was really inspired, I suppose, because this area has emerged in the last, it's 5 to 10 years, especially the last 5 years, as a very exciting aspect of immunology. And my lab and Dave's lab, we've done lots of work. I think Dave got into it ahead of my lab, I must admit, when he was in Dundee. My lab began doing this in 2010, 11 time, really ramped up in the last 5 or 6 years. I would guess most of my lab now is doing stuff on metabolism. So, and then there's huge amount of literature, there's interesting discoveries being made. Lots of labs actually in immunology have switched into metabolism, and in Infinity ourselves, by the way, I think there must be 6 or 7 labs now. We're looking at metabolic aspects of immunity, and it's unusual. I might start with that because immunologists would avoid metabolism. Often they chose immunology to get away from biochemistry, strange as it may seem, and all those metabolic pathways that you'd be familiar with. It didn't seem that relevant to immunology, in truth. Now, there was always an area to do with things like diabetes and heart disease and the immune system and insulin resistance, and TNF could drive insulin resistance, for instance, and there was links into type 2 diabetes from cytokines. That was kicking around for a long time, actually, and very interesting stuff there. And it's still happening, by the way. And then there was also kind of stuff on glycolysis in macrophages. But we all thought, if I go back 10 years, this is just to do with energy and ATP and biosynthesis, given that that's the main function of metabolism. What's happened is, in a single sentence, metabolites that were previously only understood in terms of standard, say, bioenergetics or synthesis or catabolism, the usual things you would have all learned in second year, I imagine. They've gone well beyond that now into very specific functions in immunity and the role of specific metabolic pathways in immunity and specific metabolites. And then the bottom line would be, it's been rediscovered kind of because it was going around decades ago. And new technologies like mass spec and metabolomics, and I'm going to mention a lot of metabolomic stuff actually, has really helped here. And my lab has made a couple of reasonable discoveries in the area. One on succinate, which is in Krebs cycle, and one on itaconate. There are 2 big ones. We also had a couple of big papers on a thing called PKM2, which is in glycolysis, and that turns out to be a key regulator of Th17 cells and activated macrophages. So it's interesting. What's happening is, I suppose, we're merging 2 fields, standard metabolic pathways into immunology and discovering really specific things. I guess the surprise is we found some very specific events that you wouldn't have expected in certain metabolites governing. There's a word for you now, actually governing the phenotype of the immune cell. So if you look at metabolites inside an inflammatory M1 macrophage, it's very different to an M2 macrophage. Or if you look in Th17 cells, it's very different to Tregs. Or if you look at NK cells, which Dave has done a lot of work on, pioneering work there, when you activate an NK cell, you get interesting metabolic changes that aren't there in the resting state. And they seem specific. And there's a lot more work to be done. I'd recommend this as an area for you to get into because we're just beginning. It's a challenge. It does involve a lot of biochemistry, actually, so you need to master some of that stuff. But it's really interesting. And then what I'd say as well is it's giving rise to potential new therapeutics. My big passion, as you all would know by now. Can we exploit these discoveries for therapeutic gain in immune and inflammatory diseases? And my company, Citrix, another one I founded back in 2016, they're going after itaconate and PKM2. That was discoveries made in my lab, and that's a great example of discoveries in my lab then translating into a company, and the potential then to make new therapeutics is there. So let's give you sort of an overview. And what it's about really is, what I'm going to give you now is 2 lectures. It's kind of like research seminars, so get ready. This is the sort of stuff I give when I used to be on the road in the good old days. So get ready. It could get a bit technical. And remember, just to re-emphasize, anybody who has any questions, don't hesitate to email me and I'll get back to you because I'm here to help. And anything unclear, please email me. Now, it's kind of about this, where you have to break through the efficacy ceiling. Now, as you may remember from other lectures I've given to you, I hope, we have treatments for rheumatoid arthritis, that's RA, inflammatory bowel disease, multiple sclerosis, and there's anti-cytokine therapies and so on. The trouble is, you reach what's called an efficacy ceiling, and the patients might respond 50%, which is still better than nothing, remember. Some respond very well, some respond not at all, some stop responding. So we have a real need to get even better medicines for these diseases. And the standard targets are cytokines, as we know, anti-TNF and rheumatoid, but you do reach an efficacy ceiling. Same in Crohn's disease. Now, there are T-cell targeted therapies like abatacept, there's B-cell targeted therapies like rituximab, and these will work, but they're targeting what you might call, we would now look upon strangely as traditional immune targets. So to go beyond that, I've been saying this for 2 or 3 years, other people say it as well, you got to go beyond these standard immune targets to breach that efficacy ceiling. In other words, try something else. It's almost as sophisticated as have another shot on goal. How do we do it? Well, metabolism, interestingly, is a prospect, which is quite different from these other things. I gave you NLRP3, those who listen to those lectures, as a great target for driving cell death. This word stroma is interesting. That means targeting cells that aren't in the immune system traditionally, like fibroblasts, for example. Very interesting stuff happening there in rheumatoid arthritis. Endothelial cells, in other words, non-traditional immune cells. They're the ideas, and many drug companies are focusing on these. Roche, GSK, for example, they're going down these roads of trying to target these different things to breach that ceiling. And that's the overarching goal, is to get brand new therapies, really. And what I'm going to explain to you now and justify is, if you target metabolism, you may well break that ceiling and get a much more enhanced, efficacious drug. In some ways, one way to think of these inflammatory diseases is metabolism has gone awry, and it's gone kind of into a strange state, a different state from the non-disease state. Can you correct it back to normal? And can you achieve remission? The big goal in these diseases is to put people into remission when the disease goes away. That can be done through metabolic targeting. And that's the goal of all this, by the way. And I'm going to give you a couple of examples of how that might work out as we go through the data. So metabolism is really good. NLRP3, we've had that already. And of course, as you will remember, those of you who did this course, I'm not sure if everybody listening to this course did the other one, but this is the 4 main pathways that I'm banging on about for these diseases. Jackson stats, which we did in spades, didn't we, in the cytokine lectures. CGAS sensing DNA, NLRP3 sensing things that are phagocytosed, and the TNF pathway. And that drives autoimmunity and autoinflammation come out of those pathways. And they're being targeted. Inhibitors are being, used to block JAKs, for instance. Xeljanz is the example there. And drug companies are developing inhibitors of the other 3, CGAS, NLRP3, and RIP kinase. And we discovered NLRP3 inhibitors very nicely, and that was what I spoke about before. But a very interesting feature is mitochondrial dysfunction lies at the basis for 3 of these pathways. And that brings us immediately into metabolism. So mitochondrial DNA will be sensed by CGAS, and if a cell gets damaged, the mitochondrial DNA is released somehow. Still, that's one of the mysteries, how it gets out. CGAS binds it, drives STING, type 1 interferons. That's been shown to happen in multiple diseases. What causes the damage, we don't know, but it's mitochondrial dysfunction. Secondly, with NLRP3, it can be reactive oxygen species coming out of mitochondria. The mitochondrial DNA can also plug in, and again, we get activation of that pathway. And mitochondrial dysfunction has been linked into cell death pathways in response to TNF. And again, that's reactive oxygen species would be part of that process. And in fact, reactive oxygen coming out of mitochondria has been known for decades to be pro-inflammatory and worked on a huge amount by people. And there are attempts to block mitochondrial ROS, as we call it, And the ROS seems to be signaling down to these signaling pathways. And that idea that the mitochondrial damage is driving quite specific events there, if you think about it for a minute. Like, imagine the mitochondrial DNA binds CGAS, or reactive oxygen species are able to activate NLRP3. There's specificity here. There'll be other targets in those pathways as well, but there is considerable specificity in those changes. And the overall kind of concept that's emerged is metabolic reprogramming. So we know various inflammatory stimuli. It could be cytokines themselves. It could be pattern recognition receptors like TLR4, which we work on a lot. It could be certain metabolites themselves will cause metabolism to change. And we use the word reprogramming because it gets reconfigured. And that then in turn will change the cell's phenotype, and you get inflammatory gene expression. Whole banks of genes begin to change that are inflammatory in response to this metabolic reprogramming event. And that in turn then can provoke autoimmunity and autoinflammatory disease. So in other words, I've got my 4 pathways. I've got 3 of them involving mitochondria, but now we go even a bigger view of it overall, metabolic change. is driving gene expression changes, and that then is provoking autoimmunity and autoinflammatory diseases. That's the idea. Still a hypothesis that we're very active in pursuing. And it's been a huge area. These are numbers of papers coming out. That was 2018. I mean, it's even higher now. You know, it takes off around 2010. And we published our first paper in 2010, I'll have you know. Not just us, other people as well. 2013, we did a Nature paper, and that was early on. And then we got a big eruption. All these papers. It's great. And another way to look at this is, here's a great book, by the way, called Navigating Metabolism by a good mate of mine, Nav Chandel. He is in UT Northwestern, and he's a real metabolic expert, and he's written a really good textbook that explains metabolism for anybody, immunologists or whoever it might be, you know. And then this is your standard metabolic pathway. And on the cover, look at this: glucose, glycolysis, Krebs, lactate, amino acids, fatty acids. That's the world we're in. And the electron transport chain, of course, and oxygen. So, but then he goes well beyond that. And at the end of that book, actually, what he has is he gets experts to speculate on the future of metabolism and where it might go next. And it's like 4 or 5 little essays. And his own essay, he says metabolism could be the driver of diseases, whereas genes are just passengers. Now, why is that important? We've been obsessed with genetics for a long time with disease, and looking at genetic variants and mutants and those genes driving disease processes. And, you know, oncogenes in cancer are a good example, but you've done lots of lectures on this, I imagine, the genetic basis to disease. It turns out the gene might be the passenger. One of my favorite movies there. Look at that. Isn't that brilliant? John Candy. Metabolism is the driver. Gene expression is the passenger. Now, what do we mean by this? Well, went to a conference a year ago when I used to go to conferences. Richard Loxley, very famous immunologist. Genetic variants do not explain the increased incidence of many diseases. Okay? So all these inflammatory immune diseases, the incidence has gone up a huge amount in the past, say, 30, 40 years. That's too fast for genetic change to be driving those diseases. So there's something beyond genes. And for example, asthma allergy is a great one. Cannot be explained by genetics because it's been too fast. So what has changed is not our genes, but our diet, environment, and behavior. Those 3 things have changed, and they're now provoking disease. And let's say the dietary change, that might drive asthma, you never know. There could be some toxin in the environment. Our behavior, that means not taking enough exercise, whatever it might be. Now, you might have a genetic component there, of course, that's sort of is predisposing you, but it's the environmental, dietary, behavioral change that reveals the genetics. The great phrase, nature via nurture. You know what I mean? In other words, you might have a certain nature, which means your genes are in a certain state, or you may have certain mutants, but then when you nurture it through things like diet or environment, then you see something emerge. So you wouldn't see the disease just because of genetics alone. You superimpose on top of that other influences like diet and environment and behavior. And the reason why Nav emphasizes this, metabolism is poised to sense these modifications. Of course it is. Because diet, you know, that affects metabolism. There could be stuff in the environment altering metabolic pathways. Beta amyloid building up in your brain, whatever, you know, that changes metabolism. And then exercise. And of course, it links into gene expression then causing disease. So understanding metabolism then might provide therapeutic targets to alleviate these increases in diseases. And it could actually be about epigenetics. And that's a very interesting concept. And I'm sure you've done lectures on this. I hope you have. Because what's happening here is, it's not so much the genetic gene expression part as such, in terms of, say, a change in a promoter in the DNA that might change a gene, or it might change the protein because of a mutation. It's not that. It's the expression of the genes. And that's driven through epigenetics. And what does that mean? Acetylation, methylation, histone methylation, DNA methylation, you know, all those modifications change gene expression. And guess what drives these changes? Metabolites. So acetyl-CoA is needed for acetylation. Alpha-ketoglutarate, which is in Krebs cycle, is a cofactor for demethylases that can demethylate DNA and histones. Succinate can block demethylases. And then what's called carbon-1 metabolism is needed for methylation reactions. So suddenly now it begins to get a bit clearer. Now this is very speculative. We'd love to know a good list of the genes being regulated and where the specificity is. But the fact is, metabolism is connecting into the epigenome. And it's epigenetic change then that might give rise to disease. So if you change metabolism, you might alter gene expression through epigenetics. And that's extremely important in this immune inflammatory process. Now these are very top line sentences. Just grab the concept that metabolism is connected to everything. You can read about it if you like a bit more, how metabolism connects to epigenetics. Now, that gives you the overarching view. If we understand metabolic change then, because of environmental influences, and read out how that changes gene expression in disease, maybe we'd explain why a rheumatoid arthritis patient has huge amounts of TNF being made that's causing disease. Maybe we'll understand why NLRP3 is hyperactive in Alzheimer's disease, because the connection into epigenetics will help us explain some of that. Now, next concept for you, they're coming thick and fast. Let's get a bit more detail. A particular type of metabolism is at the heart of inflammation. In other words, the core of this metabolic alteration in inflammation is called Warburg metabolism. And there's Warburg himself. Now, what is Warburg metabolism? He described this, first of all, in tumors. He noticed that in cancer, tumors begin to change metabolically, and in particular, they ramp up glycolysis hugely. And that was a surprise at the time, because, you know, you think mitochondria would be a better way to make ATP. This wasn't the case. Tumors mainly use glycolysis. And not only that, it's aerobic glycolysis, which means there's loads of oxygen around and the tumor is still just using glycolysis. Whereas normally you only see glycolysis when it's anaerobic. Let's say when you exercise your muscle, glycolysis kicks in, you make lactate, no oxygen, so you can't burn anymore, you can't burn the pyruvate, so you get less mitochondrial metabolism. But strangely in tumors, they decide to ramp up glycolysis. And that's a key metabolic feature of tumors. And it's been studied in detail, and people wonder, can you exploit that therapeutically? But aerobic glycolysis was a feature of tumors. And then beyond Warburg then, because he avoided this in a way, mitochondrial dysfunction happens as well. So the mitochondria go really weird. And that happens in tumors too. Now, very importantly, the same metabolic deviation, if you will, is happening in inflammation and inflammatory cells. So if you take an inflammatory macrophage, it has ramped up glycolysis hugely. And secondly, the mitochondria are not doing their job properly. And that's a hallmark then of inflammation. And you see it in T cells as well. Th17 is a good example. You see it in NK cells. You know, any inflammatory cell, really, you see this change in metabolism. And it's characterized by increased glycolysis and then dysfunctional mitochondria. Now, that can mean low respiration. It can, in the mitochondria side, it can mean production of reactive oxygen species from that dysfunctional mitochondria. And our discovery was succinate. I'll come back to that a bit later. So that's the bottom line, really, in terms of this piece. Warburg is driving inflammation, just like it's driving tumorigenesis in cancer. Now, what this means is mitochondria then, that dysfunctional mitochondrial aspect, have become front and center for this. And there was a great review a couple of years ago on the rise of mitochondria in medicine. And look at this. They compared all the publications over time for different organelles. And look at this. Around about 2013 again, the mitochondria begin to take off. And work on the nucleus plummets. Now, the main reason for the nuclear stuff is, say, the human genome and all the genetics. So mitochondria took over. ER is down here. Golgi, you're a loser if you work on the Golgi. I hope you're all laughing. That only gets a laugh in real life. Only losers work on Golgi. No, that's not true. They're very important. But anyway, mitochondria overtakes the nucleus and becomes a big organelle because of immunology, we think. Because all the immunologists begin to work on all of them. That's an exaggeration, but lots of them begin to work on mitochondria. And off we go, you know, and then suddenly all this knowledge and all these papers begin to get published. So metabolism trumps genetics was the great phrase I used when Trump used to be in power. Now, one thing that's important is as well. So in other words, the mitochondria and the Warburg effect, as it's called, are very important. Now, what's happened as well was you might measure metabolites changing. And let's say certain metabolites go up, or let's say glycolysis changes. That doesn't mean it's causative for what happens next. It could just be, you know, correlating, but not necessarily causative. So what you want to get to is how a change in metabolism causes a gene expression change. And that's a key goal in my lab all the time. Whenever we see a metabolic change or we measure an enzyme in metabolism, does it link into something causatively for that cell, for that macrophage? Mainly we work on macrophages. And this is a very important concept in science. This actually could be the central question in science. You know, can you correlate something with causing something? Okay. So, for example, smoking. You know, people with lung cancer smoke, but is the smoking causing the cancer? And that was a big controversy for a long time. It wasn't clear if smoking was the cause of cancer. It could have been something else. Maybe smokers didn't take exercise and that caused cancer. So that's a big question. Does correlation equal causation? And I want to talk about that briefly because it is a very important scientific concept. Did you know there was a great study showing that you could correlate babies being born with storks in the neighborhood? Now, that meant the statistical power, storks must deliver babies. That was just a correlation. The storks actually were hanging around the houses in the towns because the newborn babies, the houses were warmer. That's why they were there. This is in the 50s in Sweden. And that was used as a great statistical thing, that you can get a great p-value for something. It doesn't mean it's causative. And what I'm all about in my research, and many molecular biochemical oriented scientists are, you want to get the mechanism. Like if you'd seen a stork deliver a baby, that'd be the evidence you need that the correlation is linking into causation. So even though there's the correlation with storks, it is causing babies to be delivered. Nobody saw storks deliver babies unless they'd taken too many drugs. Now, that was the p-value, 0.008 for storks and babies being born. Ridiculous. Another one I like was this. Did you know there was a direct correlation between where mad cow disease happened in England in 1992 and where they voted for Brexit? It could be a mistake to jump to conclusions. Now, that's actually not true. It was a joke, you know. So some mad cow was correlating with Brexit, but that's actually not true. You know, correlation clearly means causation in this case, is my joke. It's not actually true. So there's no correlation between mad cow disease and Brexit at all. And even if there was, it doesn't mean that mad cow disease was later giving rise to people going crazy and voting for Brexit. Now this idea then links into the notion then of measuring metabolic change, and that metabolic change having effects downstream. And remember I said it's all about the environment really. Now the environment means diet, it means exercise, it means things in the environment that could be toxic. Metabolism senses that change. And then the big thing is that then links into immunity. And it also links into the microbiome, by the way. I'm sure you've done lectures on that. Very interesting area. The microbiome is really important for disease. And you get crosstalk between the microbiome and immunity, of course you do. There's lots of studies linking microbiome change into immune function and dysfunction. And that's interesting, right? And it's exercise and nutrition mainly we talk about here. Now, all that's very interesting. And in fact, that is one aspect of immunometabolism, where people look at the microbiome or look at metabolic change in response to the environment and so on. But what I'm much more interested in is intracellular metabolic reprogramming. So that's going to be my main topic. My main research is about this. And of course, inflammation then can trigger metabolic reprogramming, getting back to that concept. So this gives you the totality of the field of immunometabolism in a way. It can be about things like microbiome. It can be about tying into metabolic diseases. But it's also about inflammation now. And that inflammation is tied into intracellular metabolic reprogramming. Okay, and that's the broad concept that we're going to continue discussing here. And of course, inflammation can impact on the microbiome and the microbiome metabolism. So you have a complex network here. These are really big areas. And I would advise you to do PhDs because it's great to do a PhD. You could work on microbiome metabolism. You could work on how metabolism controls the microbiome. You could work on how the microbiome controls the immune system, and then how inflammation controls the immune system through metabolic reprogramming and indeed controls the microbiome metabolism. And very interesting stuff is coming out. It looks complicated, doesn't it? Just get the top idea here about the interlinking of all of this. Now, let's get much more specific. So now we're going to go deep into this. And what gets me most of all is signaling. And I worked on TLRs for years and IL-1 signaling. And the crux of this was, toll-like receptors, for example, will sense LPS or inflammatory factors. I was obsessed with what are these signaling pathways. We discovered some of the adapters. And the signaling pathways will cause a gene expression change, and a range of genes get changed, and that will then cause immune and inflammatory effector responses. So maybe TLR4 will drive TNF production, or IL-6. Maybe it will prime NLRP3 expression for activation and process pro-IL-1 beta. So we're always tying in signaling into downstream gene expression, leading to immune and inflammatory effector responses. But now we're looking at specific metabolites as signaling molecules connecting into signaling pathways. That's the overarching theme here in many ways. And I'm going to give you a few examples of this. Now, let's look at this in more detail now, because obviously we're going deeper. And we are talking about, going back here, metabolic reprogramming, right? Intracellular. The big finding, I suppose, that began about 10 years ago, and again, my lab was part of this, was as follows. you do get metabolic reprogramming in response to certain immune pathways. Now let's look over on the left-hand side. Now, standard metabolism, going back to the 1950s and 60s, if there's loads of oxygen, the Krebs cycle runs, TCA cycle, ox-phos is fine, you make ATP, right? If you become hypoxic, HIF gets activated. Nobel Prize last year was for HIF, remember, big discovery. It's a transcription factor that hypoxia regulates, and that will drive glycolysis because there's no oxygen, so you can't do ox-phos. You also get the pentose phosphate pathway going up, and that gives rise to rapid ATP production, can lead into biosynthesis. That's metabolic reprogramming. So normal oxygen, ox-phos, low oxygen, metabolic reprogramming through HIF, driving glycolysis, and ATP levels are maintained or boosted if you need to make lots of ATP, if you're exercising your muscle, for example, and that's a bit hypoxic. So you reprogram it into glycolysis. And then, of course, tumors, as I mentioned, they're inclined to have glycolysis in the pentose phosphate pathway and not much ox-phos going on. That's Warburg. Now, what was the breakthrough? Danger signals like LPS and TLR4 drive HIF. And we were able to map that very closely, and that HIF drives glycolysis. So immediately there's metabolic reprogramming in response to something like LPS. Who would have thought lipopolysaccharide through TLR4 would cause a metabolic reprogramming event in the macrophage to drive glycolysis very hard? Now remember, don't forget about the correlation causation. That might be going up. What does it mean? It probably partly is to do with you need loads of ATP to fight bacteria, and it's quicker to ramp up glycolysis than to get mitochondrial biogenesis. Because if you want to, even though mitochondria make a lot more ATP, to get lots and lots of ATP, that takes time for the mitochondria to be made. With this one, you just soup up glycolysis and you make buckets of ATP. So it could be a speed thing, rapid ATP generation. That may be one reason for it. Now we've got more reasons coming up. Look at this though, IL-4, a classic type 2 immunity cytokine made in response to Helminth infection. Parasites will drive IL-4. That boosts ox-phos. So if I have IL-4 here and LPS over here, you get quite different metabolic consequences for those two activators of the macrophage. This is macrophages I'm talking about, and this is mainly M2 macrophages here. They're M1s over here, which means more inflammatory. So there's metabolic reprogramming. A resting macrophage has a low level ox-phos, low level glycolysis. If you give it IL-4, you get a big increase in ox-phos. If you give it LPS, you get a big increase in glycolysis. Now that's kind of a foundational observation for this field, and both involve metabolic reprogramming. And another reason for this, and I'm looking for reasons all the time, is you might need a slow burn. Ox-phos is very efficient. It doesn't generate much ROS. It's kind of very lean burn mode. Maybe that's good for anti-parasitic immunity, just to keep parasites under control. And we live with parasites in our bodies, and we sort of have maintain them in a symbiotic way. On the other hand, if you want to kill bacteria that are going to kill you, you better ramp up glycolysis quick to get loads of ATP to fuel neutrophils and fuel macrophages to do their job to beat up the bacteria. So a rapid antibacterial response, and to provoke inflammation, which, as you know, is beneficial because it brings in all the mediators, the vasodilation, all the rest of it. So maybe it's a speed issue. You need speed to kill bacteria. You can be more leisurely with Helminths, and you need more inflammation then maybe to kill a vicious bacterial infection or a viral infection, as we'll see actually with COVID. But then you don't need that with a parasitic infection. That's one idea. But the bottom line is these differences exist, and we're trying to figure out why it is. And of course, in disease then, if the IL-4 system goes out of control, as happens in atopic dermatitis and in asthma, maybe limit ox-phos to treat that disease. On the other hand, if inflammatory diseases involve HIF and involve glycolysis and these changes, target that to ramp down inflammation. So these become deviant during disease. So again, you might target them. That's the other concept that we're very interested in. Now, back to the tumor business. Why would glycolysis go massively up? Well, it turns out it could be about biosynthesis, that you can make things from glycolysis. And the pentose phosphate pathway, for instance, you can make ribose, and all these metabolic intermediates. And this is a great study, I thought, when they stuck into oncogenes, into fruit flies. There was a model of this. RAS and SARC, two oncogenic forms. They put those flies in, this is a strange histological section through flies. And this is the green stuff is the tumor in the fly. They put them on low sugar, the tumor grew. Put them on high sugar, massive tumor expansion. Isn't that brilliant? And what's happening here is the sugar, the glucose, is being metabolized through glycolysis for biosynthesis, and you're making the tumor mass through glycolysis. Isn't that incredible? And that's how powerful Warburg is. So one reason for Warburg in tumors is to allow for biosynthesis. Very important. And again, that really says to me, this is fantastic. And the good analogy is like a building site, and you're bringing the bricks are the glucose, and then you're building the building. And glycolysis is taking the bricks and turning it into various metabolites is the idea. And, you know, if you now turn to the immune side of things, as I mentioned already, you know, really what's happening is it's a difference between anabolism and catabolism. Now, what do I mean by that? Well, anabolism means to make stuff. Catabolism means to burn stuff, okay, to break things down. And the inflammatory state is anabolic. Now, again, what's happening here is glycolysis is the feature of the inflammatory state. And you're using glycolysis to make ATP, but you're also using the intermediates for biosynthesis. And the biosynthesis can be loads of cytokines. So another reason to ramp up glycolysis in an inflammatory macrophage is to cause a massive increase in TNF synthesis. You need an awful lot of amino acids to make loads of TNF. And then you might have gene expression and mRNA is being made. So you're making lots of inflammatory factors. And this is in the case of the M1 macrophage, dendritic cells, Th1, Th17, the endothelium really ramps up anabolism. And that means a boost in glycolysis and a decrease in fatty acid oxidation actually is a feature. And then you block the fatty acids being oxidized, and you use them for lipid synthesis, you see. So this is very much about making stuff. Now, the anti-inflammatory state is breaking stuff down. It's catabolism. So you see an increase in fatty acid oxidation, an increase in OXPHOS, and these are happening now. You're burning now fats through OXPHOS, and you're burning glucose ultimately through OXPHOS as well. And there's an increase in glycolysis too. So this is the M2 macrophage, the memory T cells, the regulatory T cells. Now, activated T cells are more complicated. They have a mix of everything, really. They've increased glycolysis and OXPHOS. It's not quite as simple, but certainly overall, anabolism means inflammation. You're making lots of inflammatory factors to drive the inflammatory process. Anti-inflammatory pathways break stuff down, and lipids seem especially important. Now, lipids can be very pro-inflammatory. So, you know, if you're building them up through limiting fatty acid oxidation or making lipids, that could be pro-inflammatory. Now, why is it pro-inflammatory? Well, you might make prostaglandins. They're an inflammatory lipid. You might make membranes for antigen presentation in dendritic cells. That's an important feature there as well. Whereas if you're burning lipids, that's going to be net anti-inflammatory. You're metabolizing them, you know, and then you get rid of them, basically. This is one reason why obesity ties into inflammation, because that is a toxic lipid environment. Loads of lipids are driving inflammation and obesity. And we know obese people are slightly more inflamed. And that inflammation, in turn, gives rise to insulin resistance and diabetes. And others would have spoken about that too, I imagine. So very interesting, this idea of anabolism versus catabolism. And if you're an immunologist, you're obsessed with mTOR. That's a key immune effector pathway. You hope you know senses amino acids. That drives anabolism. So that's on this side. Whereas AMP kinase and autophagy, which means degrading things through the autophagosome, and AMP kinase that promotes autophagy. It promotes mitochondrial function in various ways. AMPK goes up and ATP is low, and it senses AMP, and that drives a pathway involving autophagy and mitophagy, the breakdown, or mitochondrial metabolism rather. And that ties into this other side, the catabolic side. So it's very simple. mTOR is inflammatory, broadly speaking. AMPK is anti-inflammatory. And autophagy is anti-inflammatory overall because it ties into catabolism. And that's an interesting concept that's emerged as well. And remember, this side, the anabolic side, the macrophage side, is all about increased glycolysis. And then secondly, mitochondrial dysfunction ties into that as well. Now, I'm going to give you some examples from my lab. I'm not going to give the T cell one, actually, so scratch that out. And I'm going to talk about the literature, a bit of a broad spectrum now on various things that I want to explain to you. And the first little story I'm going to talk about is PKM2. Now, get ready for this. This is a specific story. Now, this stuff before was quite broad. Glycolysis on the one side, OXPHOS on the other, anti-inflammatory, pro-inflammatory. Now, let's drill in a bit. I'm going to look at glycolysis now in more detail. And I suppose the overall question is, why would glycolysis be pro-inflammatory? Can we get a link from glycolysis into inflammation? And the example is PKM2. Now, here's glycolysis. There's glucose, hexakinase, GAPDH, enolase, and there's PKM2. It's the last step in glycolysis in some ways because it converts phosphoenol pyruvate into pyruvate. And PKM2 is the enzyme that does that, or pyruvate kinase. And pyruvate then can go into Krebs cycle, of course it can, and acetyl-CoA, or it can make lactate. Remember, so if you've got a normally respiring mitochondria, you'll burn the pyruvate through Krebs to make ATP. The inflammatory macrophage has no oxphos happening really, as we'll see a bit later. You've damaged the mitochondria, you get a big increase in lactate. And in fact, lactate is a hallmark of the inflammatory macrophage. It pumps out lactate to beat the band. And PKM2 is very important for that. But we made a very interesting discovery about PKM, pyruvate kinase, and specifically PKM2. Now, what is pyruvate kinase? It's the, as I say, it's the enzyme that converts PEP to pyruvate. It turns out there's two isoforms, PKM1 and PKM2, and they're splice variants. This shows how they're made from the PKM gene. You get 9 spliced into PKM1 and 10 spliced into PKM2. And PKM1 is a very active enzyme. It's probably the main one that's driving pyruvate into the mitochondria. PKM2 is less active, so it's a bit slower. And it turns out that that allows intermediates to build up to drive biosynthesis. So tumors often have PKM2. And it's a bit slower, and you can't make as much pyruvate, and you get diversion away from glycolysis for the synthesis of building blocks for tumors. Okay, and that was first shown in cancer. So it's a very interesting area then, the relationship between PKM1 and PKM2. And this is the way the ratio exists. It turns out that, you know, as I say, PKM1 is the really active form here. And PKM2 is a bit slower. But then again, it gets more complicated because the ratio of PKM1 to PKM2 is very important. But there's a second ratio because it turns out PKM2 can exist as a dimer or a tetramer. And when it becomes a tetramer, it's very good at pyruvate kinase activity, PEP to pyruvate. So it's a bit like PKM1. When it's a dimer, it's hopeless at that. So the one in a tumor turns out to be the dimer. So if I go back here, here's dimeric PKM2. All right, and it's the dimeric one that you see, and it's less active. So it's complicated, right? And it's a very interesting discovery in tumors, actually, because you get a shift to dimeric PKM2 in tumors. It's less active, and you get then a build above the intermediates. There is still some PKM1 there, but you see a lot more PKM2, the dimeric form. And it turns out this can oscillate back and forth. Things like fructose bisphosphate, which is in glycolysis, will push the dimer to the tetramer and ramp up glycolysis. That's just like a feed-forward loop, for example. But this dimeric form, it's got separate functions. It moonlights. And guess what? Low PEP affinity, non-enzymatic, it goes to the nucleus and can regulate transcription. And that was shown in tumors. Now, isn't that a fascinatingly complicated thing? Give it to you again. There's glycolysis ramping away. There's pyruvate kinase. PKM2 as a tetramer can do this. But when it's a dimer, it goes away and goes to the nucleus. And in fact, it drives HIF, as I'll show you in a minute. And what we discovered, that was extremely interesting, I think I would say so myself, if you take an inflammatory macrophage, an LPS activated macrophage, okay, you see loads of dimeric PKM2. We don't quite know how LPS drives that. That's still a bit of a mystery. But you get loads of dimeric PKM2. Now, immediately then, you know, you're kind of slowing down glycolysis a bit. Now, glycolysis still works. It's still burning away. But more importantly, the dimeric form goes to the nucleus. And guess what? Interacts with HIF and drives HIF-dependent genes. And these include genes in glycolysis like LDHA and Warburg metabolism. GLUT1 is a glucose transporter. So you ramp up glycolysis through dimeric PKM2 by boosting expression of lots of enzymes in glycolysis. And PKM1 is still there then to make the pyruvate, and then the pyruvate ends up as lactate. But if you're an immunologist, guess what we discovered? IL-1 is a HIF-dependent gene. And therefore you need dimeric PKM2 coming out of glycolysis, leaving its job, moonlighting away from glycolysis, into the nucleus, driving IL-1. Now there was an interesting discovery. So we're linking now glycolysis to IL-1 production through dimeric PKM2. Isn't that absolutely mind-blowing? And there are drugs that they were developed for cancer initially that will force the tetramer. So if I go back here for a minute, hang on, sorry. If I can get the tetrameric form, if I can make this more active, it'll increase the flux through glycolysis, right? And make this a tetramer now. And then that will decrease biosynthesis and it'll have an anti-tumor effect. So if I can force this tetramer away from the dimer, it speeds up glycolysis, less biosynthesis, and then stops this dimer doing its thing because I'm forcing the tetramer. And we could use these drugs to force the tetramer, and it completely suppressed IL-1. It also suppressed glycolysis because now you've got this decrease in expression of the enzymes in glycolysis because you need the dimer to drive those enzymes. So there we had a very interesting drug to block IL-1 production by forcing PKM2 into a tetramer. And then other labs did the same thing as us. It turns out the dimer is key for Th17 cells. If you force the tetramer, you'll decrease IL-17. That was our paper just middle of last year. So it's not just in macrophages. I'll give you another one we found was PDL1. Very interesting paper a couple of years ago now. That needs dimeric PKM2. Now PDL1 is part of the checkpoint. It stops, tumors being attacked by immune cells. And again, it needs this dimeric PKM2. So another reason these dimeric, these tetramerizers, like they're called DAS and TEP, if you force the tetramer, you'll decrease PDL1 expression. That can provoke anti-tumor immunity. We had a paper on that a couple of years ago. And then we looked inside T cells, as I said, and we got very similar results. I mean, will I show you this data or not? Hang on a minute. I don't want to kid you. Yeah, okay. Great. So we've also done stuff on T cells outside the macrophage. And I just set it Th17. Bit of data for you now. Here is PKM2. It gets phosphorylated as the dimer. Massive increase when you activate T cells. We're activating T cells with a CD3, CD28 combination. Look at this. It all goes to the nucleus. It does take a bit of time. But here we have a resting macrophage, or T cell. Activate the T cell. It all goes to the nucleus. And that was a very interesting result. And then we put on these tetramerizers, the compound that can force the tetramer. And look at this. We could suppress T cell proliferation. That was a profound effect on T cells dividing. And we could block TNF production from T cells by forcing this tetramer. Now, I'll be back to this at the end in case you're confused. It's all about the dimer tetramer. This is forcing the tetramer. It could block glycolysis because you're blocking expression of all the enzymes in glycolysis because you need the dimer to interact with HIF to drive the glycolytic enzymes. So that was quite neat. And very importantly, we block Th17s. There's a suppression of IL-17. This is from our paper, don't worry. Blocking Th17, the facts here, 26 out of 8. That was nice, blocking Th17s. It also blocked Th1s, actually interferon gamma, were blocked by this tetramerizer. And even better, it kind of turned them into a Treg. So here we see this FOXP3 as a marker for Tregs. You could boost it with the tetramerizer. Look at that. And you could do it in unpolarized cells. Th17s, we could push those into Treg state, and Th1s. So you're reprogramming the cell here. And then this is important. We did this with Kings's lab as well. We did the experimental autoimmune encephalomyelitis model of MS. And these mice developed paralysis. We gave them the TEP46, the tetramerizer. And look at this, we could delay the onset of disease. This is paralysis. So this is mice giving a compound that can force PKM2 into a tetramer and delay the days of onset there. So PKM2 may have therapeutic potential. Now here's the summary of this. Get ready. If you didn't understand all that data, just write this down. I'll realize this. That PKM2 dimer, now most unexpectedly, out of glycolysis, the PKM2 dimer drives Th17 cells. These are all markers of Th17s, these ones. And it also drives Th1 through a thing called the OMEs, not required for TBEP, but don't worry, it's driving Th17 and it's driving Th1. And look at this, the dimer inhibits FOXP3, so it limits Tregs. So now, when we tetramerize, okay, we force that dimer into a tetramer, that will block Th17s, it'll block Th1, and it'll boost Tregs because you're forcing a tetramer. Because remember, the dimer is inhibiting FOXP3. And there's all these things we found. HIF is downstream at the dimer. mTOR, c-MYC, STATs are downstream, interestingly. And inflammatory macrophage activation. So inflammatory macrophages, Th17s, and here's ones, need dimeric PKM2, an enzyme previously only thought of as being important in glycolysis, had been linked into tumors, but here we have it now, very importantly in immunity. And it's like a hub for inflammation. So there's metabolic pathways. Just think PKM2 is critical for the inflammatory state in inflammatory macrophages and in Th17s. Now, what's very interesting here is then you can take metabolic targeted drugs and get a TH0 to become a Treg. So if you block glycolysis, because you need glycolysis for Th17s, rapamycin blocks mTOR, a drug called dimethyl fumarate, back to that later, TEP46. they will all block Th17s, but they allow the Th0 to become a Treg, and we don't fully know why that is. Now, again, this is the therapeutic idea. You can reprogram a T cell then into becoming an immunosuppressive Treg. It can be very useful to suppress EAE, MS, by blocking glycolysis, by preventing that dimeric PKM2 and forcing the tetramer. And you can prevent Th17 development. So very interesting therapeutic prospects, and that's what my company is doing. And in the second lecture, which I'll come on to a bit later, I'm going to take more on this metabolic reprogramming event. But let me go back just to re-emphasize what I'm talking about here, just to give it to you very simply. If you don't understand some of it, just get the following concept. I told you glycolysis is up in inflammation. It's a hallmark of the inflammatory state. One reason is to divert intermediates into biosynthesis so that the inflammatory macrophage or the inflammatory T cell can make lots of cytokines. Okay, so there's a simple idea. You ramp up glycolysis, divert away a bit into metabolites to make cytokines, IL-17, and they make loads, so that's why you need this shift. That's one concept. But our discovery is an enzyme in glycolysis, pyruvate kinase, behaves in a very complex way. It becomes a dimer in the inflammatory macrophage that goes to the nucleus, and that will increase gene expression, slightly ramping down glycolysis to allow biosynthesis to happen. And the mitochondria are badly damaged, so you can't go all the way to mitochondria. Back to that in the next lecture. And that dimer then drives all these events. It drives IL-1, it drives Warburg, and it drives IL-17. And that's my summary here. So that dimeric PKM2 will drive inflammatory macrophages, Th17s and Th1. But very importantly, if you target these pathways therapeutically, this is the complicated thing. If you block that, if you prevent the dimer, the T cell becomes a Treg and becomes anti-inflammatory. Now, why would that be? We don't really know. We think it's sent into mitochondria, actually, that the tetrameric form can regulate mitochondrial function in some way and boost OXPHOS, you see. So, but just get this concept that metabolic reprogramming can actually give rise to a different phenotype at the end, a different cell type. And we're very much interested in what the mechanism of that might be. I will end the lecture there, and thank you all for your attention. And in the second lecture, I'm going to talk about therapeutic prospects.